Okay, Assalamualaikum. I am Anas. Today I would like to present about the diversity of marine phototroph. What is mean by phototroph? So basically it means that any of organisms that use light as their energy sources is considered as phototroph. Why we must know or understanding about this phototroph? Understanding of phototroph is very important, especially for our new biotechnology development. So in this presentation, we could see the development of technology and technique of method sampling and detection analysis. So in this presentation, we could see the method changing over the year. And then let's go to the next slide for know about it. Okay, for the primary producer, what is mean by primary producer? Okay, primary producers means that organisms that can synthesize organic using inorganic. So what the difference between phototroph and primary producer? So what makes they different is phototroph must include light source while the primary producer can synthesize ATP either using light or not. So in the traditional method, they capture the phytoplankton using plankton nets in size 16 micron and above. So they will capture phytoplankton in size between 16 micron and above lah. So that's why they only consider diatom and dinoflagellates primary productions. So in this slide, we could see the example of phytoplankton net that we use until today. So for the picocyanobacteria is mean smallest. So it's the smallest cyanobacteria that live in the marine environment. So nowadays, they are using fluorescence microscopy and accumulate knowledge of analysis had found out Cyanococcus are predominantly distributed in many environments. Then high performance liquid chromatography HPLC technique revealed that it has divinyl chlorophyll which had an absorption spectrum slightly different from that of the usual chlorophyll, which has higher 3 times. HPLC can detect divinyl chlorophyll, so it can differentiate between Pleurococcus and Cyanococcus. Between dominant, both are dominant in tropical and subtropical regions, but Prochlorococcus can be found more at tropical regions. Okay, photoheterotroph. Heterotrophic organisms that use light for energy but cannot use carbon dioxide as its sole carbon. In 2000s, 2 significant findings were reported. They are found the microorganisms that transform light energy to chemical energy in marine environment. For the number one is aerobic anosogenic photosynthesis bacteria, which have contained bacterial chlorophyll that mainly recognize photosynthesis in anaerobic anosogenic photosynthesis bacteria distribute in an anaerobic environment. For the number 2 is proteodophine that's containing bacteria. Okay, for the oxygenic photosynthesis that using far red light, in the 1996, they found a cyanobacterium that used chlorophyll D to absorb fluorescent light that called Acreochloris marina. Then in 2010, they found another red chlorophyll F from the cyanobacterium from the microbial mat on the stromatolite. At the coast of Australia, what is stromatolite? So, stromatolite is a layer sedimentary of formation that created by photosynthetic cyanobacteria. So, the example you can see at the slides presentation. So, it's on the rocks of the at the coastal. So next is for pico-eukaryotic phytoplankton in 2010 they found many amount of picohaptophytes, including many undescribed species were widely distributed in marine environment, and also found out that significant amount of significant amount of microalgae in unknown phylum in isolate phylum to date also exist in marine environments. So by using metagenome and flow cyanometry technique analysis, metagenome is basically using DNA analysis. So all it means by metagenomes is the study of genetic material recovered directly from the environment species. For the flow cyanometry technique is used to detect and measure physical and chemical characteristic of population of cell or particle. Strange phototrophic microorganisms. Many of strange phototrophic microorganisms was detected by metagenome analysis. In traditional method, they would use morphology method to recognize the microorganisms. Using metagenomes, researchers can now we use metagenome analysis that use DNA and to analyze another species. So using metagenomes, the researcher can analyze microbial diversity and also identify new proteins, enzyme, and biochemical pathways. So, in the last four conclusions, in this study we have seen that every year we could found a new species that is important for us to study due to the development of biotechnology in relations to the food web in marine environment. The study on energy flow are important and relevant to the global environment change. Preservation and protection of living marine resources and diversity of marine microbes. Thank you.